Hi, my name is Katharine Kripke. I have a PhD in immunology. I've been spending my entire career studying HIV, virology, immunology, and prevention, including 6.5 years as Assistant Director of the HIV Vaccine Research Extramural Program at NIH. And the past 10 years, I've been working on mathematical modeling of HIV prevention. Today, I wanted to talk a little bit about the immunology behind why the current SARS-CoV-2 vaccines that are available that were designed against the original variant of the virus protect against severe disease and death from the currently circulating variants, but don't protect as well from infection among the currently circulating variants. One thing to know about the immune system is that it very specifically recognizes molecules, and it has two mechanisms for doing this. One is antibody-mediated immunity. So antibodies are proteins that are secreted by a cell called a B cell, and anything that an antibody binds to is called an antigen. Antibodies bind to intact viruses in the intracellular space. So in this figure, we're seeing these purple molecules here are antibodies, and they're sticking on these proteins that are on the surface of this SARS-CoV-2 viral particle. And you can see that they're just physically obstructing the virus, making it difficult to bind to a cell so that it can infect the cell. So each B cell lineage produces a unique antibody that exactly fits the antigen that it binds to. And antibodies, like I said, physically block viruses from infecting cells, and when they do this, they're called neutralizing antibodies. Antibodies that are bound to viruses also tag them for clearance by the immune system. The other type of immunity is T-cell immunity. So this is the other mechanism that specifically recognizes molecules, and it works differently. So in this picture, which is actually a figure for flu virus, but the principles are the same, what we can see is that here's the virus, here's the antibody bound to the virus on the outside of the cell, this is the cell here, this blue part, and here's a virus that's in the process of getting into the cell. So once the virus is in the cell, its proteins can get chopped up by the cell, or here's the viral nucleic acid that's directing the manufacture of new virus proteins. So these proteins can get chopped up by the cell and loaded onto molecules called MHC molecules. And the MHC molecules take the little chopped up pieces of viral proteins to the surface of the cell, where they show them to the T cells. And again, the T cells are recognizing these viral pieces, which are called epitopes, very specifically. Each T cell recognizing a single epitope. But the epitopes are from chopped up pieces of proteins, so they're able to include the entire protein and all the proteins of the virus, not just the surface proteins. So here we see T cells are recognizing chopped up pieces of viral proteins called epitopes from inside infected cells. Each T cell lineage exactly recognizes a single epitope. And then we have two different types of T cells. We have CD8 T cells, which are sometimes called killer T cells, and when they recognize an epitope presented by an MHC, they directly kill the cell that's showing that epitope. And then some cells will present in a different type of MHC to another type of T cell called a CD4 T cell or a helper T cell. And helper T cells mobilize the immune response. They're like the generals of the immune response. They call all of the other immune cells in and activate them to clear an infection. So why do the current SARS-CoV-2 vaccines that were made against the original strain of SARS-CoV-2 protect against severe disease and death for the current variants, but less so against infection for the current variants? So antibodies are the first line of defense. They can block infection. We saw in the picture, they're actually physically keeping the virus from getting into the cell. And since neutralizing antibodies bind to intact proteins on the viral surface, not chopped up proteins, the virus only needs to change the outer part of its surface proteins in order to evade antibody responses. And some of the new variants can evade antibody responses, such as Omicron. So in this case, if there's an evasion of the antibody response, then the virus infection can be established. On the other hand, since T cells recognize these chopped up parts of viral proteins inside cells, they can respond to all the parts of all the viral proteins. So the T cell response is more diverse, and therefore it's harder for the virus to get around it and evade it with mutations. There's just more T cells seeing more different things. But since T cells only recognize viruses inside infected cells, the virus has already established infection before the T cells can effectively mobilize the immune system to fight the virus. So the T cell response is slower, but it's more resistant to mutations. And that is why these vaccines protect against severe disease and death, because they've got the T cell response to get rid of the virus before the virus progresses too badly, but less so against infection. That's all for today. Thank you.